Hello again and welcome back. Thank you for joining me today. Today, we will discuss matrix metalloproteinases, or MMPs, as promising targets for therapeutic intervention. This is the 8th installment in our series on emerging drug targets. If you are new here, please consider subscribing. We release new videos every Saturday. In this episode, we explore recent advances in understanding the roles of MMPs in disease pathogenesis and how these insights are translating into novel therapeutic strategies. So let's dive right in. 1. Scientific overview. The matrix metalloproteinases are a family of zinc-dependent endopeptidases that play pivotal roles in extracellular matrix or ECM remodeling. MMPs regulate tissue repair, angiogenesis, inflammation, and metastasis by degrading the structural proteins that form the actin, such as collagen, elastin, and fibronectin. Aberrant MMP activity is implicated in cancer progression, fibrosis, neurodegeneration, arthritis, cardiovascular diseases, and aneurysm formation. In general, dysregulation of MMP function is associated with pathological conditions involving ECM degradation and deposition. For clarity, there are several subgroups of MMPs, including the collagenases, gelatinases, stromalysins, matrylysins, and membrane-type MMPs. As an example, the collagenases such as MMP1, MMP8, and MMP13 break down fibrillar collagen, while the gelatinases, MMP2 and MMP9 target denatured collagen and type for collagen. MMPs are tightly regulated by tissue inhibitors of metalloproteinases, the TIMPs, and their dysregulation is frequently observed in chronic inflammation, metastasis, or vascular remodeling. 2. Real-world evidence. Accumulating real-world evidence, or RWE, supports the causal roles of MMPs in disease pathophysiology. In neurodegeneration, longitudinal brain imaging and cerebrospinal fluid, or CSF, biomarker studies have linked MMP9 activity with increased blood-brain barrier permeability in Alzheimer's disease and multiple sclerosis patients. In oncology, high levels of MMP-2 and MMP-9 in tumor biopsies correlate with poor prognosis in breast, colorectal, and pancreatic cancer. In fibrosis, RWE from liver biopsy repositories shows elevated MMP- in progressive NASH, suggesting roles in ECM turnover and fibrotic evolution. In cardiovascular disease, clinical data from coronary artery disease, or CAD, patients shows MMP-9 as a strong biomarker for vulnerable plaque and myocardial remodeling after myocardial infarction, or MI. 3. Therapeutic modalities targeting MMPs. Given their central roles in disease pathogenesis, it is not surprising that MMPs have been targeted using a wide array of modalities. First-generation direct inhibitors like 𝗪𝗪𝗪stat and 𝗪𝗪𝗪stat showed some early promise but failed in later-stage trials primarily due to dose-limiting musculoskeletal toxicity associated with off-target effects. Second-generation inhibitors like 𝗪𝗪𝗪stat and 𝗪𝗪𝗪stat were more selective but also proved ineffective in late-stage trials. However, several novel and next-gen modalities are showing great promise. These include monoclonal antibodies targeting MMP-9 and MMP1 with isoform-specific binding, small molecule inhibitors, MMP-cleavable prodrugs, nanobodies, and RNA-based inhibitors like siRNA and antisense oligonucleotides. For example, the anti-MMP9 antibody 𝗪𝗪𝗪𝗪 by Gilead is currently in phase 2 trials for ulcerative colitis and gastric cancer. Gene editing tools like CRISPR-Cas9 have shown good preclinical results for MMP modulation in invasive glioblastoma and metastatic models. Dual-target biologics combining MMP inhibition with anti-angiogenesis or checkpoint blockade strategies are also showing good preclinical data in oncology. 4. Clinical pipeline and trial landscape. As of mid-2023, there are 11 active or recently completed trials leveraging these various modalities to target MMPs. These include Gilead's anti-M 𝗠𝗘𝗫𝗧 antibody 𝗠𝗘𝗫𝗧 in ulcerative colitis and gastric cancer. AstraZeneca's pan-MMP inhibitor 𝗧𝗛𝗪𝗓 in COPD and acute lung injury, and an MMP-9 𝗠𝗛 from 𝗡𝗘𝗫𝗏 for age-related macular degeneration, or AMD, among others. Notably, the majority of ongoing trials employ antibodies, antibody-drug conjugates, or nanobodies, indicating a renewed interest in selective and locally activated MMP inhibitors. Several academic groups are also pursuing first-in-class MMP cleavable prodrug platforms that are selectively activated in diseased tissue to avoid systemic toxicity. 5. Market landscape and competitive positioning. Despite earlier setbacks with broad-spectrum MMP inhibitors, the next-generation approaches are reactivating this target class. The interest in MMP inhibition is exemplified by Gilead Sciences' acquisition of True North Therapeutics for up to $400 million in 2019, reflecting its long-term oncology interest. AstraZeneca's 𝗧𝗛𝗪𝗓, a potent oral MMP inhibitor, has seen renewed attention as part of its respiratory pipeline in response to COVID-related acute lung injury. Several academic startups are pursuing platform-based MMP cleavable prodrugs and nanoformulations to treat fibrosis and solid tumors. Additionally, venture capital firms are increasingly interested in fibrosis-related MMP programs, especially in combination with anti-transforming growth factor beta or anti-IL-13 agents. One company, Tenaya Therapeutics, raised over $100 million to develop an oral MMP inhibitor for the treatment of pulmonary fibrosis. 6. Recent sales and financial activity. While no MMP targeting drug is yet approved for market, financial indicators suggest a rising strategic focus. Gilead's investment in MMP antibody platforms, including 𝗠𝗘𝗫𝗧, was estimated at $400 million, including development milestones. AstraZeneca's respiratory pipeline, including MMP inhibitors, contributes to its $2 billion annual investment in inflammation and fibrosis research and development. Licensing deals for localized MMP prodrug activation tech have emerged in early-stage oncology partnerships with undisclosed terms. Academic startups like the Antibody Consortium at the University of Oxford are securing significant venture capital funding, highlighting the potential of MMP-targeted antibody-drug conjugates. In the last 24 months, venture capital funding in fibrosis-targeting startups using MMP inhibition has exceeded $150 million. 7. MMPs in cardiovascular disease and emerging indications. Emerging evidence suggests that MMP2 and MMP-9 are key modulators of post-myocardial infarction remodeling, ventricular dilation, scar formation, and fibrosis. They are also associated with aneurysm progression via aortic wall degradation and stroke via increased blood-brain barrier permeability and cerebral edema. MMP inhibition may reduce infarct size in stroke models, suggesting therapeutic potential. Novel strategies targeting MMPs are being explored for acute coronary syndrome, heart failure with preserved ejection fraction, HFrEF, and atrial fibrosis. 8. Strategic implications. The clinical translation of selective MMP inhibition is gaining ground through 3 key strategies. First, advanced delivery systems like liposomes, antibody-drug conjugates, and nanoparticles allow for targeted drug delivery, reducing systemic exposure and side effects. Second, combination therapy is a prominent theme with co-administration of MMP inhibitors with immune checkpoint inhibitors, VEGF inhibitors, or anti-fibrotic agents. Third, biomarker integration, such as monitoring MMP-9 serum levels, is increasingly used for patient stratification and treatment monitoring in fibrosis and oncology trials. In conclusion, the field of MMP inhibitors has evolved significantly since the early setbacks with broad-spectrum agents. Next-generation strategies employing advanced delivery systems, combination therapy, and tailored biomarkers are driving renewed interest in this target class. As our understanding of MMP biology deepens and new technologies emerge, we can expect to see more creative and effective approaches to harnessing MMPs for therapeutic benefit in oncology, fibrosis, cardiovascular disease, and neuroinflammatory disorders. Thank you for watching. Please subscribe and comment below to share your thoughts on this emerging target class. And we will see you next week.